Howdy, this is Zia Barton, presenting genetic and external isoamyl acetate production in Apis mellifera. Apis mellifera is the European honeybee. It is one of the most common types of bees in the world, prevalent to Europe, West Asia, and Africa. So isoamyl acetate is going to be the focus of this presentation. We're going to be talking about what it does on this slide. It goes by several different names, and what it does is it is the alarm pheromone for bees. Basically, it alerts bees to danger, letting them know when bees have sensed something either smelled or visually that is dangerous to them, like a predator. It causes aggression or defensiveness, which can be monitored in like respiratory behaviors and agitation, movements, that sort of thing, exploratory behaviors, such as going after things that they see or smell. It can also be a repellent in high amounts, but an attractant in low amounts. It is released from what we usually see as the stinger, but is also called the sting of a bee. And even if the sting is removed from the bee, the pheromone still lingers on it. So say if a bee stings a bird and the bird flies away, but the stinger's still in the bird, that IAA will still be on the bird because it is attached to the stinger. So some general factors would be defensiveness. It's going to be genetic factors. So heritability of aggression, that's what that means is really that bees can inherit certain factors of aggression. So like take African honeybees, for example. Those are going to be more aggressive than your typical bumblebee or European honeybees like we're talking about. So obviously that is a heritable quality. They are genetically predisposed to be more aggressive than other types of bees. The number of worker bees is also a big impact. If a hive is larger, they're going to send out more bees when there is danger alerted to them. Behavioral phenotypes. So this kind of goes hand in hand with that Africanized honeybee, that kind of thing. Certain bees are more prone to being aggressive. We call those bees usually guard bees or soldier bees. Sometimes it's their job to be more aggressive than others. And age. Studies were done on JH, that junior hormone that is in bees when they are younger. The less junior hormone that is in them, the more aggressive they become. And the experiment was that they injected JH into older bees and they became more gentle and less reactive. And some environmental factors, pollen quality. So if a bee has more pollen, they're going to be more gentile. They're going to not need to defend it as hard. If they have less pollen of less quality, they're probably going to be more aggressive because they've been searching for it. They have to defend it a lot harder if there's less of it, that kind of thing. Pollen distance as well. If bees are further away from the hive, they're less likely to be alerted when there's danger. So that would make them less defensive. Colony intruders, if they are more likely to have intruders, they're probably going to be a more defensive hive. So say if they get attacked regularly, they're going to be more defensive than bees who have never been attacked or don't have any predators around them. There's some other things that affect bee defensiveness. The amount of SAP or SAP, that's that stinging alarm pheromone. So basically when IAA is produced, it alerts the bees to the fact that there is danger. And then when they get agitated, SAP is produced. So they're linear. They go together. One affects the other. If IAA is produced, they're more likely to generate some SAP too and be more likely to sting. Bees are also genetically predisposed to stinging. Like I said, that Africanized honeybee, they're more dangerous than European honeybees because they are more reactive. External stimuli. Stinging is influenced by the things that a bee can see, smell, and touch. So if a bee sees something outside of its hive, it's going to start generating some IAA. If they smell something, if they smell other bees generating IAA or some other hormones off of some other animals, they're going to be more likely to sting and generate that IAA. Both quantitative trait locus mapping and behavioral studies show that guarding behavior is a key factor in colony stinging response. That guarding behavior is, like I said, that alerting of the IAA. The more bees that you have that are reactive, that are watching out for danger, the more likely you are to react to external stimuli and become more aggressive. So number of guard bees is also important. Guard bees is a specific job that can be done by certain bees. They are like little soldiers. They stand as lookout. And in a phenomenon called recruitment, they can release IAA, a lot of it, or, a decent amount to call other bees to them in like a reinforcement kind of thing to help them against any danger. And the more guard bees that are called, the more likely that they are going to produce sap and stinging will occur. Our real main question for this research project was, do genetic factors or external factors produce more isoamyl acetate in Apis mellifera? So really we're going to judge this based on guarding defensive behaviors, and IAA production is really hard to measure, so it's more like IAA that is produced during guarding behaviors. So this first thing here, this first figure shows the number of guards versus the type of hive that they are in. And this was during an experiment where they took bees that were raised in a gentle hive, less defensive hive, and put them in a more defensive hive, and vice versa, and bred them in. So this is not the first generation, this is the second generation. And we really see that when you're in a defensive hive, you become a defensive bee. You're likely to have more guards, be more reactive. And if you're in a gentle hive, you're going to have less guards, be less reactive. So I know this seems like it's more of like the environment. If you place a bee into a more aggressive environment, they're going to end up more aggressive. But it's more about the fact that they were bred with aggressive bees. So the overall kind of message that I got from this article was that the differences observed in the number of guards were due to the genetic composition of the colonies, and that the variance components indicated that phenotypic variation in the number of bees that were guards for at least one day, which is what you see here, was attributed more to genotypic effects than environmental, even though environmental were important. Obviously, if they had nothing to defend against, they would not be defensive, but that it still was due to genotypic variation and the expression of guarding behavior rather than just putting them in an aggressive environment, they become aggressive. Figure 2, this is a pretty large figure, so I broke it down into 3 pieces. Here is figure 2A. So what we're looking at here is the correlation between colony aggression and location. So this data was gathered using experimental threat stuff. What we really see here is that the closer these aggressive colonies are to each other, the more likely that other colonies are going to be aggressive. I think it's really interesting to look at 6, 10, and 9 and 1, where they smush the ones in the middle and make them a little bit more aggressive. But also interesting to note that 1 and 2, which are very close together, are complete opposites. So it does depend. It's not all about locale, but it does play some impact. And then here, relation of the colonies. This is figure 2B. It's showing that these colonies are actually related to each other. They are cousins, they have split up, and you can see by the thickness and the number of those branches between each other how interrelated they are. So the ones that tend to be, they tend to share similar reactivity. And you see the red ones are more related, they're more likely to be red, and the blue ones, even this light green colony 12 here is related to the blue, where it is also related to a much smaller orange above it, which does impact that, making it, lighter green than it would be darker blue. I think it's very interesting to note here that the relationship intensity does determine some of that, but not all. Here, figure 2C is the association of AMF frequency. So that is colony minor allele frequency versus colony aggression phenotype. Basically, you see here that these groups are linked and that the results indicate variation in aggression in honeybees, both at the individual and group levels, is significantly heritable. And as seen in other association studies of aggression-related behavior, individual genotype-phenotype association analysis is limited, but overall in the hive, it is obvious. And that is what this graph shows. And then our third piece of evidence here, guarding response. We're going back to talking about those guarding bees and such. So we're really going to talk about how the relationships between colony response alarm pheromone targets and targets for stinging has a relatively high heritability, and it was reasonable to hypothesize a common genetic basis for both types of defensive behavior based on how long they were guarding in reaction to the genetic correlation. So, in conclusion, most data points to genetic factors being more impactful than external factors. So, even though location and all of that did play a role, it was more important that they were genetically predisposed to be guard bees or genetically predisposed to be aggressive, like we showed with that figure 1, where they became aggressive by breeding into an aggressive colony. The significance here is human parallels. Can this data point back to us? Do we also have something like IAA that is more likely to raise our aggression? We can research that based on bees. And protecting our ecosystem. When we know the temperament of the bees, we can protect ourselves better, and we can protect those hives and understand them more. And then knowledge on heritability in insects, this does not just apply to bees. This applies to things like termites and ants who have similar life systems. So we can go ahead and expand on those if needed. Here are my references. Thank you very much.